Hello, my name is Camila Perea, and today I will be discussing an article called The Ubiquitinase USP9X Regulates the Invasion of Prostate Cancer Cells by Regulating the ERK Pathway and Mitochondrial Dynamics. I was interested in this article because my uncle has prostate cancer, and I wanted to understand a little bit more about the pathway and the scientific background behind it. This article deals with the ERK pathway, which we see ERK right here. So essentially, when growth factor is bound to the RTK, it becomes phosphorylated, it dimerizes, and is able to phosphorylate other things such as this fellow right here, which is GRAB2. GRAB2 contains a domain called an SH2 domain, and that domain will bind SOS, which is right here. Once SOS is phosphorylated and it's active, it is able to exchange this RAS GDP to RAS GTP. So once RAS has the GTP aspect, it becomes activated. And now it is able to regulate RAF. RAS phosphorylates RAF and makes it active. Now, RAF is active and it can phosphorylate MEK. Now that MEK is phosphorylated, it can phosphorylate ERK. Now that ERK is activated, it can phosphorylate over 100 substrates, and when they're phosphorylated, they can actually enter the nucleus. Here we see some examples. AP1, CMYC, CFOS, CJUN, and all of this leads to G1 to S, and it leads to all these things. Now let's jump a little bit into the background of this article. Starting with a little background on prostate cancer, it is quite common. It is the second most frequently diagnosed cancer in men, and a cause of the mortality, a big cause of it, is its invasive nature and the metastases that it causes. This highlights the importance of early diagnosis and treatment due to its very aggressive nature. Ubiquitination is a very important regulator. It is a post-translational regulator, meaning this affects and modifies proteins after they've been translated from mRNA. These enzymes serve many important roles. Some of them include cell cycle control, DNA repair, amongst many others. And the specific enzyme that we're dealing with today is USP9X. Now let's talk a little bit about the abstract. This enzyme has been studied in several other cancers, but there's not much investigation in prostate cancer specifically. And in this current paper, they're investigating the expression and the significance of this enzyme in prostate cancer when compared to normal tissue cells. It actually finds that the expression of this enzyme, USP9X, is downregulated in these tissues, the prostate cancer tissues, compared to healthy tissues. It is also thought that this increased expression is associated with local invasion. And also the depletion of this enzyme is associated with the upregulation of different things such as MMP9 and the phosphorylation of DRP1, which will be important later on. There's also a significant increase in ERK, which is actually an activator of these fellows right here. So what they did essentially is that they investigated the ERK pathway, they inhibited it, and saw exactly what role it played with this enzyme. One last important thing to know about this USP9X enzyme is that using target proteins that would cause proteasomal degradation, it is able to remove things such as malignant tumors, which would be very useful for cancer patients. If you'd like a quick review about how ubiquitination kind of works, essentially you have a protein of interest and something binds to it. That's usually some sort of enzyme. Here it says that it targets protein towards degradation. So some sort of enzyme that is directed by USP9X. Once these are bound together, it causes a tail of ubiquitin to form. And once this is kind of marked with this tail, it heads its way to its fate in the proteasome. And it gets degraded. Okay, now onto the figures. A through D are some staining results. The top two are normal tissues. Bottom two are cancerous. Let's compare these two first. In A, we see strong cytoplasmic staining for USP9X, meaning there's more of it. Not necessarily that it's active, but there's more of it in these normal tissues. Here we see a difference, less of the staining adhered, so meaning there's probably less USP9X in the cancer cells. Now let's look at the Western blot. Let's look at F here. This, by the way, is the loading control. We're comparing normal and cancerous tissues. So initially in these, we do see a presence of protein, of USP9X, and compared to cancer, there's actually a decreased amount, at least in this one. This one maybe looks a little bit similar to that one, for example, but there is a decent decrease in cancer cases 1 through 3 compared to 1 through 3 of the normal cases. In the following cases, we see kind of the same trend right here. About 3 of these show a significant decrease in USP9X protein when compared to the normal tissues. So that is pretty significant. Here we quantify the relative amounts of this protein. In most of the cases, USP9X protein is decreased in the tumor. The tumor is the non-shaded. So, for example, here, 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 and here. So in 5 out of the 8 cases, there's downregulation of USP9X in the tumor. So why exactly is this significant? This is significant because if we go back to the initial observations, we see that they believe that there is a decrease in USP9X expression in prostate cancer. And why is this an issue exactly? Well, it is an issue because we learned that it has demonstrated to do things such as removing malignant tumors. Therefore, if there's a decreased amount in the cancerous tissue, there might be a connection between that. That may be one of the reasons why these tumors are continue proliferating and continue to exist, because there is less amount of essentially the disposal mechanism of these types of cells. Now let's look at figure 5. Here we see 2 different cancers that they evaluated. They're looking at things such as ERK, AKT, P60. The 2 lanes are one that is control, and the other one which they used a method to knock down this enzyme, or they knock down this protein. So essentially they're taking away its function. I believe that's what knocking down does. So let's look at the differences between these two. Now let's compare these 2 lanes. For ERK protein expression, they seem to be quite similar, but the phosphorylation is a little bit different. Here we can see that when the USP9X is there and it's doing what it needs to do, ERK is slightly less expressed. There's a difference. There's not much difference in phosphorylation of AKT or phosphorylation of P65. Here we can see in the other cancer the same thing. The band is kind of smaller in the control than it is in the knockdown. One can also say that in the absence of USP9X, ERK is upregulated. Here they sort of quantify this. We can see that phosphorylation of ERK is really the only thing that got affected by the knockdown. Here, these things remained pretty similar. Now in B, we introduce not only the knockdown, but also this PD98059, which is actually an ERK inhibitor. What does this mean? When this is positive, ERK phosphorylation should be downregulated. So let's go through lane by lane. Lane 1 is essentially the control. Both of these are negative, so we'll use 1 to compare. And then we look at lane 2. Lane 2 has the knockdown. So we can see that USP9X is downregulated in this one, and ERK is upregulated, and which we had already seen here. Whenever this knockdown is introduced, ERK is upregulated. So we see that again. Another thing we see is MMP9 upregulated, which we had read about in the abstract, and as well as the phosphorylation of DRP1, which we also read about in the abstract. In lane 3, we see that there's no knockdown, but we introduce the ERK pathway inhibitor. So here, USP9X is nice, it's present, it's expressed, but then the phosphorylation of ERK plummets, and this is clearly because it's being inhibited. There is ERK, so it's not that there's just an absence of ERK, it's just being inhibited. It's not a downregulation of ERK, it's just being inhibited. We also see a possible correlation between ERK, MMP9, and the phosphorylation of DRP1. Since ERK has many substrates, these are probably two of its substrates, because when there's a lack of phosphorylated ERK, there's a lack of MMP9, and there's a lack of phosphorylated DRP1. Here we see similar results. These are just two different cancers, all right? So let's go through this. This is again the control. Here we have the knockdown of USP9X. We can see band gets a little thinner, so, and also phosphorylation of ERK is upregulated with when this knockdown is added. This is a good constant that's going on, as well as MMP9 and DRP phosphorylation. They're both increasing because there's an increase in phosphorylated ERK. And then right here we see that there's no knockdown, but there is ERK inhibitor. And when there's ERK inhibitor, USP9X is upregulated. Of course, ERK phosphorylation is plummeting again, even though there is ERK. So that's good. And then MMP9 is slightly thinner. I would say it's much more obvious in this cancer, but you can kind of see the difference in thickness. And then same with phosphorylation of DRP1. These are the corresponding graphs for what I just explained. But yeah, this is kind of the general idea of the relationship between ERK, USP9X, and then these two ERK substrates. To conclude, we learn that this enzyme USP9X is downregulated in many cancers, including prostate cancer, and we know that this enzyme is involved in ubiquitination, which essentially works as the activation of garbage disposal mechanisms per se, meaning it is what causes certain things such as malignant tumors to be taken up or absorbed and destroyed, degraded by the proteasomes. So without this, that would probably cause a more aggressive and invasive and metastatic tumor. So it could probably contribute to all these characteristics. And this was tested by seeing what the lack of USP9X would do in correlation to ERK and in correlation to ERK substrates. And we see that initially in this graph, we see that there is a pretty significant difference between normal tissue and cancers in the level of USP9X in the stainings. And in the Western blots, the protein produced by USP9X is downregulated in cancer. And then we see here that when the knockout is applied, the ERK phosphorylation is upregulated. And when ERK is downregulated, there's a good response in the USP9X. It is upregulated when there is. If there's anything that these Western blots are alluding to, is that there's definitely a relationship between the phosphorylation of ERK and this enzyme, as well as these two substrates of ERK. Thank you so much.